Fantastic. Welcome, everyone. It's great to see so many people here today. Welcome to the next seminar for the ASC Training Centre in Predictive Breeding. We'll be hearing from Mark for today. And just briefly, so I'm Maddie James. I'm a research fellow in the ARC Centre of Excellence for Plant Success in Nature and Agriculture. I'm based in the School of the Environment, and for the people on campus, I'm over in the Goddard building. And my work really is looking at the genetic basis of complex traits in nature. I'd like to start by acknowledging the traditional owners upon the land on which we meet, which are the Yugara and Turrbal people here in Meanjin. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. Just a bit of housekeeping for today. So today's seminar is scheduled from 1 p.m. to 2 p.m. And then after the presentation, we'll have questions and answers. During the webinar, for the people online, submit your questions and answers in the Q&A tab, not during the chat function. And today's seminar will be recorded as well. So it's my absolute pleasure today to introduce Professor Mark Cooper. Most of you will know him, of course. So Mark has spent over 30 years of working in crop breeding, both in academic and industry roles. He's a Deputy Director of the ARC Centre of Excellence for Plant Success in Nature and Agriculture. And his main research interests combine theoretical, simulation and experimental work. And he really uses prior knowledge of trait genetic architectures to help design breeding strategies so we can have more accelerated crop gain and genetic improvement for quantitative traits. And so one example of this is him using crop growth models in combination with genomic prediction to account for the impact of genotype-by-environment interactions. And today we'll be hearing from him about long-term genetic gains and additive models as well. And he's titled it Back to the Future, so I'm hoping to see some references to the Back to the Future movie, but no, maybe we'll see. So thank you so much, Mark. Let's give a hand for Mark. Thank you.
Thank you very much, Dr. James. It's certainly a privilege to be introduced by you. Greatly appreciate it. Thank you, everybody, for coming. Hopefully, I'll be able to do justice to the title, and maybe there will be some sort of throwbacks to the movie, so forth. So I will get started. I, too, would like to acknowledge the traditional owners and really appreciate their significant contributions. And if you've ever spent some time looking at this piece of art, it's actually quite significant. And interestingly enough, the trajectories that sort of are travel through time that are indicated by the trees below are pretty indicative of some of the themes that might actually come out in this presentation. So there's a real synergy between the principles, the ideas, and the art to sort of help you to think about things in a bit more of a different multidimensional way. And so hopefully I'll be able to pick up on that theme and stimulate some of your thinking. So what I'm going to do is talk about, and it's basically an introduction to a publication that I've been involved with developing, and it's work that's spanned sort of from the 1990s through to present, where the theory and the experimental work have synergized with each other. The work started, I'll use the laser pointer on here. So it started with Dean Podlick back in the 1990s, when we were looking at questions such as, what if you had the whole genome sequence of a species that you were breeding with, and you could get markers across the genome, and you could do this for a whole breeding population? How would you go about breeding if you had access to that information? So we were developing prediction, sort of simulation platforms around which we could look at these types of prediction questions, using the knowledge that we had today to think about where would we like to be sort of 30 years into the future and so forth. It might sound a little bit crazy, because in the 1990s, people would say, well, we're not interested in what you're going to be doing in the 2020s. You might say, I'm very interested in what we're doing in the 2020s. And you may be able to look back in another 30 years. I probably won't, but you'll be able to look back and think, well, yeah, maybe there were some questions we should have thought about back then that might have helped us to think about where we should be in 30 years' time. So this is sort of an interesting piece of work that then connected with the original work at the University of Queensland with Dean, as we moved into the private sector and connected with Frank Technow, who I first met at Hohenheim University, where we were starting to talk about some of the ideas with individuals there. He was interested, joined the team, and started to look at alternative algorithms for simulating a wide range of different genetic models. So the Back to the Future paper, so Back to Future 2 that I'm talking about here, sort of was a co-authorship with Dean and Frank. And so just to throw back to the movie, yes, there is going to be a trilogy. We're working on version 3. We've been contracted. We've got the actors on board, and we're working pretty hard on it right now. So it would be pretty interesting to talk about that paper by itself, but what I was really more interested in was the connections between what we were doing here and work that I've been doing recently with Daniel Ortiz-Barrientos in the Center of Excellence, where we've been looking at if you could build a picture with all of these resources I just talked about of the landscape that a breeding program was trying to navigate, how could you use that description and describe properties of the landscape to help you to navigate the future a bit better than we're currently doing? And the property we're going to talk about, or I'm going to talk about, are additive channels. And I'll sort of try and explain to you what additive channels are. They're sort of a discovery that's come out of this collaborative work with Daniel, and comparing it back to some of the things we were finding when we were simulating the breeding programs. So you could think about this sort of classical picture of trying to model a breeding program where you have some baseline program and you're looking at some modified technology or breeding strategy, and you want to compare that as a prediction strategy over time, and actually look at the advantage that you actually get with the approach you want to take and compare that to the baseline. You could do this experimentally. Unlikely you would do that in a crop breeding program. You may do it with an experimental organism. Our experimental work is exploring these things with Arabidopsis as a model system. Come back to that later. But it's sort of a pretty average looking view. It loses a lot of information. The picture down below, believe it or not, is also a representation of what happens when a breeding program actually moves across this landscape. So one of the things that I want you to start to think about is the response surface that the genetic models is creating, the phenotypic response surface we're trying to navigate, which is the adaptation or crop performance in your target population environments, is shaped by the genetic architecture of the traits and how the environments are changing over time. You could represent that as what we might call a landscape. People talk about it has shape, have some level of ruggedness, and it changes. So you could think about this, and some of the analogies people would use, it's more like looking at water, where the waves actually change the shape of that landscape. And that sort of is about as far as what you'll see people talk about it in the literature. But the reality is it's a bit more complex than that, in that as the breeding program operates across that landscape, it also changes the shape of that landscape. So there's a change that's actually happening such that the things that you're trying to navigate to in the future could be quite different than the things that you actually see when you look back. The critical part of this, and this is the tie to predictive breeding, is the current paradigm with the way we actually build prediction models is we use data that's generated historically by breeding programs to train our models based on the shape of that landscape as it's happened or at the frontier of what we're doing, and hope that there's some relationship between this and the future. Okay? I mean, that's the basic premise. That's the assumption that we're making, that the way we build and parameterize our models today will get estimates of the average effects of allele substitution that we can actually build our predictive models on, so we can put selection pressure on allele combinations and shift the populations over time, select improved genotypes. Okay? That all works well with certain models that we often assume. So the whole idea of the Back to the Future was looking at the past, thinking about how well it represents the future. How do you really build models at the frontier to help you to predict to the future? And just as a spoiler alert for movie 2, but not movie 3, additive channels are actually a way in which we can tune the predictive models that we're actually using to better navigate to the future. Okay, so that's the teaser for paper 3, for example. Okay, let's move on. So what I'm wanting to do is talk a bit about this paper. I'm going to do it in terms of what I find is, for me, it's the operational version of the breeder's equation that I actually have used over my career, where you're interested in response for multiple traits, small t, in the target population environments. You make a whole lot of breeding program design and crossing decisions and impose selection pressures on different traits. That's the selection intensity here. But you do that based on the data you can access today. And you build models based on these data. That's what the capital M is. You can think of it as your multi-environment trial, or you can think about it as the models you actually create to predict the future. And then we are interested in looking at building models with some level of predictive accuracy. But to be clear, those models are based on the data that you have now. So that's why it's got this subscript M. So the models are shaped based on the data you've captured, not necessarily the genetic variation that you're actually trying to use in the future. So think about that. This is sort of ahead of the frontier of the breeding program. You're building your model in the past. So what you're assuming is that there's a very good relationship, as I said, between the information that's embedded in the models, the data and the models you can create today, and the models that may be relevant for the genetic variation in the future. So that's what this term is in here. And basically, you can think of it as a correlation between the information inside your data sets or your models today with the information you need to navigate the landscape of the future. So usually the breeder's equation is written without that term in. So basically, it means everybody in the room that's doing that is assuming that that number's 1. Okay, that's basically what you're doing. Whether you realize it or not, you're making that assumption. And so what I've spent probably most of my career on, doing all of these things, but also designing experiments so that we can estimate that number. Okay, the reason you might be interested in that's easier to talk about today is climate change. Okay, population pressures and so forth. It's clear that the future is going to be different than the past. So there's a very explicit formal framework for actually connecting sort of the breeder's equation across the whole TP space in relation to what we can do right now. And what I'll emphasize is that these additive channels are pathways that you generate with the way the breeding program operates that connects how you've shaped the genetic variation today into the genetic variation you're going to encounter as you go forward over future cycles of breeding program or as you make different crosses. So what I'm really interested in is the synergy between these two papers. And all of this is based on long-term work that was done working as a commercial maize breeder, where we were able to look at the history of the trajectories that were achieved by the breeding program starting in the 1920s, sort of developing inbreds out of land races and building heterotic groups and hybrid breeding that's driven through to the current day. So I've been fortunate enough to be heavily involved in that work as a lot of these resources I talked about before became available to us. So this is the historical G by E by M context that shaped the history that you're building the future on top of. The other thing is we spend a lot of time in the sort of late 1990s, 2000s, building the capabilities to actually construct the predictive models we were going to use for the future. So the genomics resources, the environmics resources characterizing our experiments and target population environments, and the phenotyping capabilities that are becoming much more available to everybody today. So this gave us the mechanisms around which we could actually build the predictive models that we were only simulating in the 1990s, but starting to build the operational infrastructure for in the late 1990s into the 2000s, and then operating breeding programs with those capabilities. This publication here is sort of a summary of the way we did this and put it into practice and the outcomes from that for the targeted work that I was leading on improving drought tolerance of the maize hybrids that were deployed across North America and South America. And it sort of gives you a little bit of a journey of the background that I've just talked about with the experimental examples that provided all the information that we're actually using. The other paper down the bottom here is the origins of the theoretical model that actually sets up all of the models, the genetic models that underpin the simulations of the breeding program that we actually did in this Back to the Future trilogy. 2 so far, 1 to come. See if it gets cancelled, but we'll keep working on the movie. So, key discovery, I've already indicated this, that's come out of the synergy here is that, and we discovered this from 2 completely different ways. So this is sort of where it gets really exciting when you're doing research, that you get a discovery doing something one way, and it converges on exactly the same answer when you're doing asking the same questions, but approaching in a very different way. So in the work with Daniel, it was sort of very theoretical, a bit abstract to some people, but it's a lot of fun. And here it's a whole lot of simulation. A lot of the statistics we were generating were describing how the system was being linearized, which really was forming additive channels. And here is the theoretical framework for actually building metrics to describe when are you in or when are you out of an additive channel. I'll sort of introduce that. Then we can sort of look at that in relation to these ENK landscapes. And some people might not know what these are. So I will do a little bit of a quick introduction of the terms. I'm not going to get into this in detail. I could certainly spend the rest of the talk on this slide, but everybody thinks, why did I come here? And I'll sort of try to move through this pretty quickly. And this is the original algorithms for setting up this were done with Dean Podlick when he was doing his PhD. He was here with me in the 1990s. Okay. And so we were actually working with people at the Santa Fe Institute in the US that were actually exploring these graph models and network models. And we tried to generalize that for applications in breeding. So as a quick summary of the terms, where are we? Where's my little laser pointer there? So the E basically represents the different environment types with their frequencies of occurrence that make up your target population of environments. The N, you're all familiar with this, these could be all the genes or the QTL that are determining the traits you're interested in. And then the K is how a measure of how these interact. And in a graph theoretical framework, these are the edges that connect the nodes inside your graph model. So here you've got sort of a very simple toy example based on 10 nodes, 10 genes, n equals 10. We got 2 environments, environment 1 and 2. Even with the additive finite locus model in this case, you can actually get changes in levels of expression or on and off between environments to generate some level of G by E interaction. Okay, and this can actually shift your genetic correlation between environments just by changing which nodes inside that, even the infinitesimal model, are actually on and off, depending on the size of these effects. You can actually add the epistasis by then building some graphs by allowing some of these things to interact. And then there are many ways we can actually put functions, equations. We've done a whole lot of different ways of modeling these connections or conditional effects between the alleles, between one locus and another. And then this can also depend on the environment. So some genes might not be expressed in one environment, therefore that contribution to the network doesn't happen in that environment relative to another. And just to sort of make life a little bit more realistic, we were often interested in situations where we were saying, well, we didn't get to find all of the nodes anyway. We only saw maybe half of them. And so you're trying to build a predictive model based on sort of a fraction of the information you really need to build a good model. So that's the cartoon. That's the sort of basic framework of the ENK. So when I say ENK, something like this is underneath it. You can actually build this up, and we do it in a much more sort of scaled up way. So you can look at any number of genes you want and any number of edges. And so these algorithms we built were just very efficient ways of doing this. So we can actually create the landscapes and explore them. So here's an example where there's 100 nodes, and then those nodes are actually broken into modules. think of these modules as traits, okay? So there's a set of genes that are involved in one trait. They may influence more than one trait. That's another possibility. And you can have different levels of epistasis or non-additivity within one trait than in the other. And so this picture here is an example where we've got these 100 genes with this setup of interaction. And then we're only able to see 50%. Our mapping works only managed to find 50% of the genetic variation. So you can't actually see this. So you may have found some information about a couple of traits. You don't even know these traits exist, which was the reality of when we started to look at drought tolerance in maize in the 1990s. Everybody thought that more roots was the avenue to drought tolerance. 30 years later, I can say that wasn't the case. That was the wrong model. That was the wrong idiotype. And we only learned that over the whole breeding process. And actually turns out that the products we commercialized had smaller root systems, but they were much more efficient and we had to go after different traits. And those traits still resided here. And the whole idea of the iterative research was to expose those over time. And we published some of the information about these traits as they became available and we were allowed to talk about them. So everybody can say, well, graph theory, boring. Okay, I'm going to go to sleep now. Why did you do that? Well, the reality is today, with advances in all of our technologies, we can actually go to our experimental data sets. And so this is work that's been a large part of the PhD of Shunichiro Tamura, working with Mel Wilkinson, Owen, myself, and also inspired by using a lot of the prior knowledge to set up experiments with Christine Beveridge, where we can actually take any data set that you generate today, and we've got tools where we can actually construct these graphs for your data set. So if you can phenotype it and genotype it, we can do this today, okay? So you can actually build some picture of the graph structures you've got access to. So here we are. We've now got this. You can think about that. So that example that I gave you was actually for tillering in maize, because we were interested in exploring the branching network in maize. It's from a very specific cross that released variation for there. It's a very interesting story in itself, but that related back to the work by John Dobley and his team, where they were trying to look at domestication of maize from teosinte. So that was for one trait, but as I said, we can actually think about how these models connect to different traits inside a crop growth model framework, and then integrate that up to the traits we're interested in, such as yield stability, and then look at that performance over that adaptation landscape. And then from that framework, and this is work that's been published since, you can actually use the parameterization of the crop models that we were developing and parameterizing for the traits we were studying to look at how much, as we went across the continuum of environments that we were working in, how do the different traits explain the variation for yield as you move across these different target environments. So in these water-limited environments down here, you've got this trait that's having a dominant influence on the yield outcomes. As we moved right over to the favorable environments, it's got no effect pretty much, but another trait's got a much higher level of contribution to the yield variation. The nasty place in here in the middle is that most of the variation that you're dealing with is really trait by environment interactions. So there's a whole lot of conditional effects. So things, if you look at yield, might look like QTL by QTL interactions. They actually could end up being nested within a hierarchical structure of trait by environment interactions. And so that's work that was done, led by Charlie Messina and his team, working with Graham Hammer, and within the work since I've come back to the University of Queensland within the Centre of Excellence, working with David Jordan on doing this in sorghum as well. And so this gets us to this nice cartoon type picture here to actually look at what genetic gain might actually look like in a real breeding program where you're dealing with these G by E by M interactions. And I'm sort of going to come back to these earlier pictures to finally help an explanation here, tying back to the themes that we indicated at the beginning. So what I've done is I've broken the whole continuum of water availability into low evapotranspiration environment types, moderate level of evapotranspiration environment types, and high, or if you like, irrigated high rainfall type environments in the target geography. And then here you're looking at grain yield, and within each of these cells, we've got a different management strategy the farmers were looking at. This was plant population. And so we're now going to look at evaluating yield within an environment type in combination with management. So this is the G by E by M interaction. And here, what we're looking at are 3 lines. These lines represent, you can think of them 2 different ways. The one on the bottom here, so it's easy to look at this, is following this progression here. The ones on the bottom are what we would call our non-Aquamax hybrids. So you can think about these as the beginning, but the reality is these are outcomes of breeding programs that are pushing for other target environments. So they're continuing to improve hybrids, they're continuing to push them, but they're not putting as much emphasis as I was on the drought tolerance, okay? And then these other 2 lines here represent cycles of genotypes we created over multiple cycles of breeding programs. So the first one here is what we would call our cycle 1 or our generation 1 Aquamax hybrids. The second one is the generation 2 Aquamax hybrids. So we were trying to put pressure on the water limit environments. So you can actually see that we developed over the cycles of breeding a series of hybrids that did better over the range of management systems. These are just an example, but a range of management systems the farmers were using. These are on-farm experiments as well. And we can actually see that yield improvement over the cycles of breeding. When you go to the opposite, you actually see that the progress is different. So if we start with the non-Aquamax, you'll see the first generation Aquamax hybrids actually went back a bit in the favorable environments. So our target was not to lose yield potential by more than 5 to 10%. So that was our target product profile, okay? So we had to be able to provide hybrids that did a lot better in the stress environments, but also not lose in the unfavorable environment. What you will see is that by the time we got to the Aquamax cycle 2, we actually managed to pool those Aquamax 2 hybrids higher on the yield potential side. So we actually sacrificed some of the yield potential in the stress environments to actually put pressure back onto the traits that were important in the more favorable environments. Sort of a little bit of a spoiler, this was by exploiting different additive channels, different trait targets that we were going after, that we were building prediction models to guide the breeding programs towards. In the middle here, it's a bit more complex, and I won't spend time on it now, but this is where you've got to deconvolute a lot more of the trait, by environment, by management interactions to understand what's going on. But we didn't lose, so in a lot of cases we were doing as good as, but in many of the environments the farmers were encountering, we were doing a lot better by the end of this, okay? Okay, so now we got a general picture and we can start to think about this whole challenge of building models for the future based on thinking about the data we have and how we build our models using the information we have today. So we're focusing on these 2 terms here in the breeder's equation, our prediction accuracy, how well our data sets represent the future based on our current environments. And this is sort of the result of something like, we've done this billions of times, okay? So these are averages over billions of ENK models. And the bad represents the gray area of not being able to access as much of the information as bigger, okay? And the good models mean we basically managed to find more of that gray area. So that's the contrast. So you could think of them as good and better, or bad and good. The thing that I'm trying to emphasize with saying the bad is you don't have to have a perfect model. You will do better. But just because you do better doesn't mean to say you've got a great method. It just means that you've managed to catch on to some information you can use to make more predictions than you can without using that information, okay? And so basically, after doing this billions of times, and I mean, this is just something that keeps coming out of all the different algorithms we've looked at, different examples, No surprise, selection works on average. But the really interesting thing is what happens at the details. So as a breeder, you may get one roll of the dice. So you don't get to average out the result. And so what we're looking at here is if we go to these cycles after breeding for a while, this is the response difference between your two strategies that you manage to achieve for a range of models that are from sort of mostly additive, low complexity to much higher levels of complexity. So what you actually find is that the models, if you can capture the right signatures of information, can actually give you an advantage in the more complex situations. They can also accelerate and help you in the less complex situations as well. The better your model, the more information you can capture, you can lift that cloud up. So you're not the unlucky breeder sitting around down here speaking to your best friend who's the lucky breeder that's sort of sitting up there, which happens. This is what you find out at conferences. Yeah, I tried that, it didn't work. Yeah, I tried it, really worked really well. I mean, it's not, that's the stochasticity of the system. There's nothing surprising about that. That should happen. You go to conferences to share those experiences. So next, what I'm going to do, well, as I mentioned, we can go from additive. You can think about this as additive to highly non-additive or linear to highly non-linear systems. The open questions is thinking about the Back to the Future bit is how selection has worked in the past, how it will work right now at the frontier using the approaches I'm using now, how will it work for the future? So those are the questions that we're often trying to frame around these simulation experiments and how we predict into the future. So we'll pick one example. So this is one ENK model with one set of equations that have been parameterized on the edges for the nodes, and it's actually based on a real biochemical reaction. And it sort of sits around about here, and it generates some level of additive variation. And we're going to look at this response over cycles of running a breeding program. And what I'm going to do is think about the additive variation that you can actually see when you build your model, say at cycle 10. So life looks pretty good. Everything's going up. World is pretty additive. You can build models. You're predicting really well. This is what's in front of you in this particular scenario. So the question that we had when we looked at this for this particular, and we had a whole lot of these pathways we were trying to manipulate, hormones, biochemistry, so forth. How can you tell where you're going to go from the information here? And it's really hard, okay? At this point in time, we didn't really have any great way of doing that. We could run the simulation and say, well, okay, this is what happened. But we couldn't say, using the information I have today, how do I get to here rather than here? And if you got to here and didn't know about this, you might say, well, I made progress, so it's really good. So you still made genetic gain, but you haven't realized the potential of the opportunity. And that was the question we were really interested in. The thing about these things is we could see them, we could describe them from any of these models, but we didn't have a framework of saying, yeah, okay, what are they? And what Daniel came up with was a really nice development of methodologies. And we called these things additive channels. They're linearizations of the system where additive models work very well. They work well within the channel. They don't necessarily work well as you go across the channels. Okay? So this gets you, that's all the background, and that's why we would do Back to the Future and the simulation. And so what we built starting in the 1990s is we had simulation models of the whole pioneer maize breeding program. And we were running these all the time using all the information from mapping traits and studying the history of the breeding program. So if you go to this paper, it basically gives you a simulation platform that allows you to set up scenarios that are representative of our maize breeding experience. So you can build your base population, you can form heterotic groups, and it is a hybrid program. So we're building heterotic groups, and then you can decide how you're going to let the breeding programs operate. So you can have one big centralized program, with one breeder running everything. Or you can actually split it apart and allow the resources to be spread amongst different numbers of breeders. Okay, and you basically allow them to operate as different agents searching the landscape. And they sort of have rules of how they interact. And the rules that we put in place were the rules that actually were how the breeding program actually operated over cycles of breeding. So we actually were simulating the breeders as agents using the rules of building and sharing germplasm. So that was the next part of here. You've got your heterotic groups. You've got these different breeding programs. This is an example of the way you can look at that. And as these breeding programs progress, they decide how to share the germplasm they're creating, largely based on how well that germplasm is predicted to perform. And you do this on the female side and the male side. You're making crosses within the heterotic groups and making hybrids and evaluating performance across. And then continually, you're building models to say how good is a parent to be used in these crosses. And so this is what this is down here. We sort of sort out the parents in terms of their value inside the breeding program. The next thing you have to do is say, well, okay, we're interested in how are TPs changing. So this is where we started to look at climate change type scenarios, or some other government policy might come in and say, you're no longer allowed to use nitrogen fertilizer, which may seem crazy, but it could happen. It's happened in other geographies, and it could happen. So we're actually starting to look at the questions using models that represent the genetics of the traits we think are important for these environments. And so in the publication, we just took a couple of examples where we allowed something to simulate a climate change scenario. You can see that there are 12 environment types here, and that's the example that's in the publication. We're looking over 30 cycles of the breeding program, and this intensity here indicates the frequency with which each of the environment types turns up in your TPE, and therefore in your experiments as you sample the TPE over time. And so you're breeding back here, but your TPE is shifting to be more like environment type 12 rather than environment type 1. Or you think, yeah, things aren't changing, going along really good, breeding's really easy, world's additive, I've made it additive, and I'm going to keep driving it as is. All of a sudden, the government comes in and says, yeah, you're no longer allowed to irrigate, the aquifer's dried out, stop irrigating, you're growing everything dryland now. That's happening, okay? And limitations on water and nitrogen use are happening in large parts of the geography that I was working in and other geographies around the world. So you can then start to ask questions about, well, what's the shape of this landscape that we're actually looking at? And so what we would do is we would look at each scenario. This is a case where all of the models in the paper were run on n equals 500 and k equals 6 example here, took a sample of 250 genotypes and then just evaluated them from something like 300 parameterizations of the model and looked at how correlated they were between the environment types. And they're ordered so that the way we built them, and you can see that in the paper, the higher the number, the further the way that the distance, the number difference tells you how far apart they are in terms of the genetics, like I showed before. Basically, you end up with families of these curves where you get a diminishing of the genetic correlation between what you're seeing now and what you can actually expect to see in some of these other environment types. And this is, again, at one cycle. So the frequency shift, the shape of this can change quite a bit. This is just an example. The other thing you can do is you can look at the average performance of the genotypes you've sampled in one cycle to future cycles. So it's sort of the lag between cycles. And again, for the two different scenarios, the gradual and the sudden change, this is how the correlation drops in terms of your ability to predict to the future based on your current data sets. So the further ahead you're trying to breed for, the less relevant the models you're using today to predict can be for those environments. Okay? So that is sort of the setup. This is sort of like a major result, what you can see over the cycles. If we look at scenarios where we're looking at the absolute performance of the best hybrids that are produced out of each cycle, what happens to the improvement in performance from your starting point in the gradual change TPE and the sudden change TPE. And you can sort of see that, yeah, the world falls apart a little bit usually when that sudden change actually happens. The programs that are doing really badly, it doesn't seem to affect them that much. You know, you think, yeah, you're back with me again, so welcome back, and we can all start again. But I mean, these are sorts of things. There's just an amazing level of detail you can get into in here. The interesting thing, and there's another graphic I'm going to use that you can actually see on here, is that when you start off with the additive models, the large centralized program does extremely well. Okay, but when you go to highly epistatic models with a lot of G by E, the centralized model starts to struggle. Okay, whereas the distributed models, where you've got multiple programs searching different features of the landscape, actually does better. And it's not that surprising that all of the large commercial programs, that's how they actually operate. As a breeder in the region I was working in, I was working as part of a team of 5 breeding programs, all going after the same geography. We all had access to the germplasm. We were all doing slightly different things and sharing the germplasm. That's exactly how we operated. So it's not a fiction. This is exactly why we did this. The other thing that we could actually then look at is a lot of metrics. Again, for looking at additive through to highly epistatic, high K scenarios, is look at the levels of additivity, or the amount of the genetic variation that's explained by things like general combining ability. And what is the genetic correlation between programs as you go across cycles, as epistasis is built up and the different programs have searched different parts of the genetic space. So what you see is, yeah, in the completely additive model, not that surprising the way we set it up, that it stayed additive the whole time. But what happened in all the other scenarios is the programs operated to make the system that they were operating in, the germplasm they're working with, more additive. So they basically were reshaping the genetic landscape by changing the genetic variation they were working with, that then made the system more additive to build the predictive models. The interesting thing you will actually see is that unless they were sharing information and germplasm sufficiently frequently, they could lose connection with each other and they would not be able to transfer germplasm. But if they were sharing enough germplasm, they could keep coordinated enough that the improvements one found could be transferred to another. And that's all this intermediate space you get here. So we can sort of build, and this is sort of a graphic, an important graphic that sort of summarizes the results. We're looking at the cycles of the breeding program here. We're looking at levels of K. We're sorting out the ranking of the breeding programs. The one that's performing the worst is red. The one that's performing the best is yellow. And then we're looking at a series of programs that worked independently versus those that were sharing germplasm at some different levels of frequency versus those that were sharing after each cycle. That's the distributed program. And then the large centralized program. And what you can see, it's sort of a nice graphic. This is for the sudden change scenario. And I'm just going to flip to show you, and this is what I'm going to talk a little bit about, is this is the gradual change. The general picture is the same, okay? There's some details that are different, but the general picture is the same. And so there's a lot of detail in here, but one of the key things that I can show you is that the only time that the large centralized program works well is in the infinitesimal additive modeling scenario. Whenever we modeled conditional effects due to simulated epistasis or G by E, one of the other programs could outdo the large centralized model, breeding program, okay? And so this was what we were interested in, was starting to understand how these distributed programs were finding pathways to improvement that the large centralized program wasn't actually finding. And so this is where the additive channels connection actually comes in. So working with Daniel, we take the same models and we start to build statistical descriptions of how epistasis or the interactions affect the conditional, how they affect the average effective allele substitution or locus, or the value of an allele in combination with all of the other alleles. And so this is sort of a basic introduction. So you can go to the preprint that's out there now, and this basic view of the additive channel. And so what you've got here is a landscape, just like what we were generating with the ENK models. You've got a breeding population here indicated by the ellipse, and it's being moved over time on a trajectory up this actual landscape. And we're using selection to restrict the amount of variation it's dealing with to stay inside that additive channel. So the selection intensity is part of this process of controlling how much variation you're exposed to. So this is where the use of the Taylor sequence expansion actually helps you to start to describe this landscape and describe these additive channels. So the Taylor expansion, I'm sure, like all of you have encountered at some point in time, you probably thought, yeah, that was interesting. Glad I survived that lecture. And for me, I mean, I just sort of thought these things are absolutely fascinating. And basically what they do is they describe the shape of a response surface. They emulate it without necessarily having to know all the detail. And so what you can do is approximate from a starting point where you're at, call it the mean of the population, what happens to the slope and the curvature of the space with a first derivative and a second derivative as you move your population. So when you move an individual away, how linear is the slope or how non-linear is it? And that's what these terms are here. You've got this, the framework, the mean of the population, the slope. These are just like your normal breeding value effects that you estimate. We're just estimating them in a different framework, and then we can look at the curvature of the shape, okay? And then what you can do with these terms is you can analyze this space and actually calculate what we're calling an additivity index, which tells you how much of the space you're occupying, your population's encountering with the data you have, that's smooth and is not too curved, so the additivity index goes up. And so what you can start to do is think about different ways in which, as you expand your crosses and think about other features that you're familiar with in a breeding program, what happens as you move out of these additive channels by trying to make wide crosses to find more diversity, okay? Great, you find more diversity. There's a lot of non-curvature in that if epistasis is present. Natural outcrossing. So basically, there's a whole lot of these mechanisms that we can now study to understand how this additive variation is created and how we can actually translate it into a response to selection. So what this picture was here before basically was a map of where different additive channels were that we were selecting for and building prediction models to help us with specific traits. And then that allowed us to actually understand how to put pressure on traits here, hold on to them, and then build on those and actually put pressure on these other traits to build over time improvements in the trait combinations that resulted in these things with improved stable performance over time, rather than trying to do it all in one cycle. Okay, so significance of additive channels. The other thing that just point out is that this framework actually integrates gene discovery with the whole predictive breeding framework we're actually familiar with. So if you've got your favorite gene or your gene sets and you want to know how valuable they are for a prediction, you can actually calculate that within this framework. It's an emergent property. It's a trait in itself, these additive channels, and we can mathematically describe it. It's got coefficients. I can map it. I can map additive channels now in genetic space. So it's a way of actually calculating informative latent variables about selection trajectories rather than traits. And so we can actually map the genetics of selection trajectories, which gives you a basis for tuning your predictive models to stay in them or to jump between them. And so with Daniel, we're working on different metrics we can use to say how far could you jump given the underlying genetic architecture we know. They can be mathematically defined. We use Taylor series integral approximations and create these additivity indices to map the additive channels. So in a soon to be published in a significant journal, there will be a paper coming out that actually will give some access to this with experience from maize and sorghum breeding, with working with David Jordan. And coming back to my picture. So this is a breeding program navigating a landscape. So what you're doing, so now start to think about your breeding program, not as independent of this landscape, but it's actually trying to predict what it's going to encounter. You can think that there's lots of techniques that you use in a boat to navigate what the sea is going to be like in the future, but it's also creating a past. It's creating an additive channel that you can actually follow. The other thing is, when you've got multiple breeding programs, they create different configurations of these additive channels. They could be adjacent, they could be beside each other. So the interesting thing you can start to think about is, how do you design a breeding program and how does it reform the shape of a landscape to actually accelerate trajectories on that landscape, rather than just saying, can I get high predictive accuracy to accelerate genetic gain? So that's the synopsis. So this goes back to picture. As I said, we're trying this in sort of model systems, working with a team, working with Arabidopsis, and also working with David Jordan on this to think about how to do it in sorghum. Just for fun, you all love Pythagoras' theorem. I mean, people say this is so abstract, but I mean, it's just a geometrical application of things you're using every day. I mean, your mixed models are just very applied sort of Pythagoras' theorem. I mean, sums of squares to actually get integrals under certain shapes is exactly what you're doing with mixed models. You apply shrinkage on confidence you have in the information, but that's what you're doing. You're orthogonalizing the information. I mean, Taylor series approximations are just an extension of this, okay? They're not that difficult. And I know Steph said I got a mistake in my equation. I don't, but basically it's the same thing. You're trying to say, what is the shape based on the information I have of the space I'm navigating? Can I capture metrics that tell me what I'm going to encounter in the next few cycles of my breeding program? How often do I have to retrain my data set? So leave it at that.
Wonderful. Thank you so much, Mark. I think it's really interesting how, these populations can kind of traverse different additive channels to get to different peaks, and whether we can still kind of, jump between peaks. I think is really interesting. We'll take now questions from both online as well as in the audience. If you have a question in the audience, just raise your hand, but wait for the microphone to come to you so everybody online can hear you. We'll start with an online question then, and then we'll come to you in a second. From an anonymous attendee, it's assumed that with recurrent selection, genetic gain would plateau as genetic variance is exhausted. However, as the long-term selection show, genetic gain can be sustained. So how does that relate to what you shared today? Feel free to stand up here.
Actually, this framework explains that very well. The infinitesimal model itself actually has problems with this, because what you can show is that we should always be exhausting that variability in closed recurrent selection programs, is the other assumption that goes with this. But this actually shows why, with these rugged landscapes, that you're not exhausting that genetic variation. And that's one of the things that we learned very early on when we were sequencing the maize genome and resequencing across many genotypes and looking at this over the history of the program. We weren't exhausting genetic variation. So there was really the assumption about our models, or sort of the things we started to ask. The important thing to remember about any model is the map is not the territory, okay? And if you believe that the map, the way you construct your view of the world, is the territory, you're likely to end up like one of these videos where people follow Google into rivers, Okay, so it's a beautiful question, and that was what motivated us to think a lot further than the models that people are currently using to look at long-term gain and genetic variation. Thank you, Anonymous.
Hello. I was wondering, how do you figure out the right number of agents and the right amount of variation in each agent to get those channels?
So that's really a wonderful optimization question, and that's part of the next phase of research that we're actually getting into. So in Shunichiro's thesis, we actually started to use a theorem from mathematics called the diversity prediction theorem that actually not only thinks about reduction of errors and improving prediction accuracy, but it looks at the diversity of information of the models, and you can start to match that to the genetic diversity and the features that you have in the prior model. So that becomes an optimization concept that we want to look at. And Shunichiro, actually, in the last chapter of his PhD, talked a bit about some of those ideas.
Am I allowed to pop in a question as well? And then we'll go to the back. Oh, wonderful. I was just wondering how you modeled whether it's possible to kind of like jump between channels. Is it based on like reproductive incompatibilities and how you expect they kind of like manifest over time?
Yeah.
Or yeah.
So Daniel is very interested in reproductive incompatibilities, as are you. So I think that motivates some of the questions that Daniel's looking at. How far apart do they have to get, and in which traits?
Yes.
And which specific components of those traits? So you can, that's where having that prior knowledge moves this from a theoretical concept. Can you build the models explicitly around that, and how far apart would they have to get on certain things? And could particular breeding strategies hold on those traits? Or does selection hold on those traits versus others? In a breeding program, we're very interested, and that was exactly the design principle around how much interchange of germplasm is necessary to be able to get individuals in your population to a high enough frequency that their breeding value worked inside the additive channels. Can you take a novel inbred and reconfigure it to bring in some novel genetic variation through some pre-breeding work to contribute breeding value into the additive channel? And we're doing some other crazy things that you can do in simulation by just saying how far apart can you have to get, and could you jump channels?
Do maybe one last question before 2 p.m.? Yes.
Okay, thanks for the presentation, Mark. Fantastic. One other thing that contributes to long-term selection response is new mutations. Is there a way of incorporating new mutations in this framework, or are they just too random to incorporate?
No, there definitely are, Ben, and actually that's some of the things that beyond mutations, what we're actually really interested in is if you've got some real knowledge of the key nodes in the networks you're working on, we can actually target editing on features of the network to tune that variation. So when we were building our transgenes for some of the traits we were working on, once we'd found a gene and manipulated the promoter, we'd usually go into a big combinatorial process of trying to tune that. So you can think about it, I mean, it's basically this concept of programmable plants. And so how do you actually do that? And actually in a week and a half, I'm going to Canada, which is a sort of a workshop that's specifically on building research plans for programmable plants around these types of concepts. So it's not, I mean, if you just leave it to random, you can build it into your framework. It just diminishes the rate at which things change, and it builds residual variation. And there's a chance element to that where it happens, but you can target that and introduce it as well. So we did a lot of interesting things like inserting a transgene and breeding with that transgene in a population and comparing what happened to the trajectory with and without that transgene in maize. And we published on some of that work. We're also doing that in Arabidopsis using some key mutants to perturb some of the genes in Christine's branching network, for example.
Great. We'll just have one final question from online. There's a question saying, do you think using haplotypes will help improve the additive channel properties?
It will if you're talking about cis regulation. But if your network is trans epistasis, haplotypes won't help you. But that's where linkage disequilibrium and holding on to recombination and selecting for combinations of genes across the genome is the way that you would actually deal with this. And it's also why we're interested in LD, because the rate at which some of these preferable complexes get formed across the genome and within chromosomes actually puts selection pressure on the amount of recombination you would want to have in a chromosome, and how you would select combinations of haplotypes across chromosomes. So you can certainly use that framework, and it sort of does allow you to actually think about some of the actual selection units that selection's actually working on. So it's definitely an area we're very interested in working on.
Fantastic. Thank you so much, Mark. Given it's just past 2, let's give Mark another round of applause. So for the people who are in the room, please feel free to stay, have some snacks, and stay until 2:30 or so. You can continue chatting to Mark. Thank you for everybody who joined us online. Just a reminder that these seminars are recorded, so use this QR code to look at past seminars as well. And yeah, thank you everyone for attending.